Welcome back. We are going to get into the concept of muscle tissue now. We're going to go down into the microscopic anatomy and the molecular level to see basically how muscles are going to contract. So, first of all, we're going to be looking at what's called a muscle fiber or a muscle cell. So, this means exactly the same thing, cell and fiber. These are very, very long cells. So if you look at a muscle, so think about biceps brachii, the cells are the length of the muscle. Okay, so they are the length of, the muscle body in there, so kind of like your upper arm area. The longest muscle in your body is called sartorius. It runs from the lateral hip to the medial knee. So it's the one that runs diagonally across the thigh. The cells of sartorius are the length of that muscle. So they're over a foot long. Okay, so these are really long cells. Now, they started embryonically as multiple cells that fuse together to form these very long cells. So you're going to find multiple nuclei. So all of the nuclei are still present. So these are what are called multinucleate cells. The cell is surrounded in the plasma membrane, which is called the sarcolemma, and it's filled up with what's called the sarcoplasm. This is not terribly different from the plasma membrane or cytoplasm, just has a special name because we're dealing with muscle cells. Now we're going to have a lot of mitochondria in here because these are incredibly active cells. There's going to be a huge demand for ATP, and we know that mitochondria are the powerhouse of our cell. We're going to have what's called the sarcoplasmic reticulum. It is this blue network that you're seeing here. This is a modified endoplasmic reticulum, and its job is to store, release, and uptake calcium. So calcium is going to be a big player in muscle contraction, and it's going to be stored in what we call the SR. At the ends of the SR, it's going to expand to form what are called the terminal cisterna. So you can see it here and right there. Between two terminal cisterna, here and here, is going to be what's called a transverse tubule. So there are going to be openings on the sarcolemma that lead into these transverse tubules. So here you can kind of see one without anything in it. It would be attached up to the sarcolemma, and then whatever happens on the plasma membrane, which is going to be an action potential, it's going to be an electrical current, that's going to allow, the T-tubules are going to allow that signal to dive very deep into this cylindrical cell so that all of these get the signal, not just the ones around the outside. So we're going to have a terminal cistern, a T-tubule, and then another terminal cistern, and those are going to form what's called a triad or a triad. So potato, potato, doesn't matter how you say it. We're going to have what are called glycosomes, and myoglobin in here as well. These are kind of storage units for the muscle. Again, super active cell. Glycosomes are going to store glycogen. And we know glycogen is a polymer of glucose, and it is a short-term energy storage. So we need to have some glucose in the muscle cell that can be made available to be fed into the mitochondria to make ATP. Myoglobin stores some oxygen in the muscles. Again, this is to drive ATP production. The mitochondria function better when we've got oxygen available for them to utilize. So all of these things, the multiple mitochondria, the glycosomes, the myoglobin, this is all driven by the fact that this cell requires a huge amount of energy. Now you got a lot of stuff in there, but the majority of what you've got inside the cell are what are called myofibrils. So that's a myofibril, that's one, that's one, that's one. It's like 80% of the volume of a muscle cell is taken up by myofibrils. And myofibrils are going to be this very highly organized arrangement of what are called myofilaments. and they're organized into structures called sarcomeres. So here you can see a sarcomere. Okay, so here's running from here to here. So a sarcomere is the contractile unit of muscle. This is what actually shortens and then allows the muscle to contract. You can see that they run back to back to back to back. So all along the very long length of a muscle cell are collections of these sarcomeres that are running back to back to back to back. Now, each sarcomere begins and ends at a Z disc. You may also see this referenced as a Z line, same thing. So you're going to go from Z to Z within a sarcomere. Now, we said that the sarcomere was an arrangement of the myofilaments. So we have two different types of filaments. We have thin filaments represented by blue, and we have thick filaments represented by red. We're going to divide the sarcomere up into regions based on whether there are thin, thick, or both types of filaments present. The H zone is where you find only thick. So running right through here, you can see there's only red filaments running right through here. The M line is this line of proteins running right down the middle of the H zone. What that's doing is cross-linking all of the thick filaments together. The A band is the area of overlap. So here we've got blue, red, blue, red, blue, red. So this is the A band, runs from here to here. And then the I band is where you find the thin filaments only. And you can see that an I band spans two sarcomeres because there is a Z disc in the middle of them. So if we look at a cross section through the different regions, we'll be able to really kind of see this very hyper-organized structure. So we'll start over here in the I band. We do a cross section right through here. What we're going to see is thin filaments only. And you can see that they're not random in their arrangement. They have a very specific arrangement within the I band. If we go through the H zone, you're going to see thick filaments only. Again, not randomly placed in here. Very organized structure. If you go right through the M line, you're going to find the same arrangement of those thick filaments, but you're going to see all the cross-linking proteins holding them together. And then if you go through the A band, you finally see the thick and the thin filaments together. You can see that each thick filament is surrounded by thin filaments. So there's absolutely no randomness when it comes to the organization of a sarcomere. So let's talk about the filaments for a second. We have thin and then thick filaments. Now, they aren't just single molecules. So let's start with the thick. It's a little bit more straightforward. The thick filament is made of myosin molecules. Each myosin molecule looks kind of like a double-headed golf club. So you've got two tails that wind around each other, and these are proteins, and then two globular heads here. These heads are going to be able to interact with actin on the thin filament. So we got an interaction, point of contact between them. A thick filament is made up of hundreds and hundreds of these myosin molecules. So you can see you put them all together here, and that's where you get your thick filament. Now the thin filament, okay, so here's your thick, and now we're going to talk about the thin. Okay, this is the area in the middle. This would be in the H zone where there's no myosin heads. It's called the bare zone. M line would be running right down the middle of that. The thin filament is thinner, smaller, but is actually made up of more different types of proteins. The primary protein is globular actin. So each one of these red balls is going to be an actin subunit. And if you want to picture how this molecule looks, you would want to take something like a beaded necklace and then twist it together. Okay. Now, each one of these actin subunits has a binding site for the myosin head. And if the binding site is open, the myosin heads will bind and the muscle is going to be contracting. We don't always want our muscle to contract. So we have a protein called tropomyosin, this long filament here, that actually lays over the top of the binding site. So it is a physical block to the myosin heads binding. The troponin complex, this little guy right here, is going to be the regulator of tropomyosin. This is going to determine if tropomyosin is blocking the sites or if it opens them up. And it's going to do that by interacting with calcium. So that's where the calcium is going to come into play. And remember that calcium is stored in the sarcoplasmic reticulum. So you start putting all of these pieces together here. So your thin filament is actin, tropomyosin, and troponin. We also have some connector proteins as well. So the end of the sarcomere is right here. This is a Z-disc, and you can see the thin filament is directly attached. The thick filament is not a direct attachment. There is an elastic protein called titin that is going to attach the thick filament to the Z-disc. The thin filament also has a connector. It's called dystrophin. This is a protein that is going to allow the tension or the force generated in the contracting sarcomere to be transferred out onto the sarcolemma and then into the actual muscle as a whole. You have to be able to connect the force from the sarcomere to the muscle. And to get the idea of how important that concept is, this is the protein that is affected in muscular dystrophy. And so when you think about what muscular dystrophy does and the fact that the muscles are incapable of generating force, you can see how very important something like this one protein is. Sometimes it's really hard to grasp something that seems so abstract, and why would we make you learn each individual protein here? But there are significant consequences to these proteins not working. So dystrophin is associated with the thin filament, and it transfers the force out into the muscle as a whole. So this is going to show you kind of a hierarchy. So you start with a muscle made up of fascicles, which are made up of muscle fibers, which are made up of myofibrils, which are made up of sarcomeres, which are made up of myofilaments. So make sure that that concept, that hierarchy, makes sense to you, because as we move forward and we get into how muscles contract, you really have to have all of this solidified. So this is going to be a little preview of how muscles are going to contract. It's called the sliding filament model of muscle contraction. Here we're looking at a relaxed sarcomere. So here's our Z-discs. We have the I band here, the A band, the area of overlap, and then in the middle here is our H zone, and that's our M line of cross-linking proteins. When the muscle is stimulated to contract, what is going to happen is the actin is going to slide over the myosin. So these ones are going to move in this direction, and these ones are going to move in this direction. So the Z discs, watch them, are going to get closer together. And what happens is that the H zone disappears. The area of overlap becomes the entire area. So it's going to be the interaction between the actin and the myosin that's going to allow that sliding to occur. So you want to make sure that you're really good on all of this terminology and you understand what makes up the thin, what makes up the thick filaments, what makes up a sarcomere, before you move on into where we're going next, which is going to be the actual nitty-gritty of how muscles contract.